It begins in a chamber no wider than a grain of sand, hidden deep in the fallopian tube, where light never falls and the outside world feels impossibly distant. Inside this narrow corridor of fluid, the first 24 hours of human existence unfold with a precision that borders on impossible. No heartbeat, no consciousness, no form. Yet the choices made in these opening moments will echo across an entire lifetime. We often imagine fertilization as a single spark, one instant, one collision. But the truth is far stranger and far more intricate. It's not an event at all, but a sequence of meticulously timed biological negotiations where every molecule, every ion channel, every membrane shift plays its part with quiet, unforgiving accuracy. Before the day even begins, the sperm have already been moving for hours, threading through gates of cervical mucus, navigating swirling chemistries, dodging immune defenses designed to destroy them. The egg, suspended in the ampulla, waits in a state of poised incompletion, holding the final step of maturation hostage until the right signal arrives. And when one sperm finally closes the distance, the entire landscape changes in an instant. Electrical gradients flip, enzymes detonate, membranes harden. The egg awakens from a cellular sleep that began before birth. This is the first day of life, a realm where identities are drafted, chromosomes meet for the first time, and a single cell prepares to divide into two. Here, invisible to every eye, the nervous system's earliest whisper emerges, not as neurons, but as a choreography of ions and energy, a proto-rhythm that will one day become thought, feeling, memory. Silent, precise, relentless. Now let's step inside that darkness and watch the first day unfold. Inside the fallopian tube, the first 24 hours of life do not simply happen. They unfold through a cascade of cellular negotiations so exact that even a fraction of a second out of sync could end the entire story. This is biology at its most cinematic, quiet, internal, and breathtakingly orchestrated. The egg, resting in the ampulla, is more than a cell. It is a suspended decision. Its cytoplasm holds mitochondria inherited from maternal generations, ribosomes waiting for instruction, and a meiotic program paused for decades. The egg's surface is protected by the zona pellucida, a glycoprotein barrier known as ZP1, ZP2, and ZP3. To most cells, this would be impenetrable, but to one sperm, it becomes the gateway to possibility. Meanwhile, the sperm that survive the journey undergo capacitation, a neurohormonal transformation driven by ionic flux. Calcium and bicarbonate rush inward. Protein modifications strip the membrane of cholesterol, making it more flexible, more reactive. Even here, the nervous system's logic is mirrored at a microscopic level. A heightened state of readiness, like neurons reaching threshold. The sperm's tail shifts into hyperactivated beating, each whip-like movement powered by mitochondria coiled in its midpiece. As sperm reach the egg, chemical gradients tighten like an invisible funnel. Progesterone, released by cells in the corona radiata, triggers bursts of calcium inside the sperm, guiding its final approach. There is no conscious awareness here, yet the choreography feels intentional. Microsignals aligning two cells that have never met. When one sperm binds properly to ZP3, the acrosome reaction detonates. Enzymes spill outward, carving a microscopic tunnel through the zona pellucida. But this is not brute force. The egg chooses whom it will allow close. Only sperm that bind in the correct biochemical pattern can trigger this reaction. It is biology's first safeguard, a subtle form of selection built on molecular recognition. As the sperm pushes through the zona, its movement grows erratic and intense, hyperactivation at full power. Ion channels along the sperm membrane flare open, pulsing with calcium waves that fuel every thrust. Inside the egg, microvilli on the oolemma prepare for contact, stretching outward like tiny hands. When membranes finally touch, everything changes. Fusion activates a rapid electrical shift, a depolarization across the egg membrane, a fast block to polyspermy. Within seconds, cortical granules beneath the egg's surface release enzymes that modify and stiffen the zona pellucida. The gate slams shut. No other sperm can enter. The egg now commits to a single story. This instantaneous surge of calcium inside the egg travels like a wave, igniting metabolic awakening. Dormant proteins activate, translation surges. Mitochondria reorganize to supply the massive energy demand that is about to begin. Only now, after decades of waiting, does the egg complete meiosis. The final separation of chromosomes forms the female pronucleus, a structure luminous with tightly packed DNA. Meanwhile, the sperm nucleus swells as its chromatin loosens, shedding proteins suited for fast travel and adopting those built for stable inheritance. It becomes the male pronucleus. Microtubules extend like tracks laid in real time, drawing both pronuclei toward each other. They drift slowly, silently. Gravity does not guide them, but biochemical gradients do. This is the closest thing to a nervous system this early stage possesses: directional signaling, intracellular highways, checkpoints ensuring everything stays precise. When the pronuclei meet, they do not merge immediately. Instead, they pause, side by side, membranes dissolving, chromosomes aligning on a shared spindle. This is the moment the new genome assembles for the first time. Not symbolic, not poetic, literal. 46 chromosomes, 23 from each parent, stand shoulder to shoulder, ready for synchronized division. Mitotic machinery ignites. Cohesin proteins hold sister chromatids together. Kinetochore fibers pull. The spindle tightens. And roughly 24 hours after fertilization began, the zygote divides for the first time. One becomes two. The two cells, blastomeres, are perfectly matched, identical in content, but unlimited in potential. They do not grow, they subdivide. Cleavage divisions produce cells smaller and smaller, each carrying the full blueprint of a future human. Each capable, for a brief window, of becoming anything: placenta, skin, neurons, heart muscle. The zona pellucida remains intact during these first hours, keeping the embryo compact and preventing premature implantation. As the two cells become 4, then 8, cytoskeletal networks shift continuously beneath the surface, arranging the blastomeres into a tightly packed sphere. Ion channels pulse rhythmically, managing pH and energy distribution like the earliest hint of an internal regulatory system. This is not yet development in the organized sense, but it is momentum. Raw, relentless momentum. By the end of the first 24 hours, the embryo has not traveled far down the fallopian tube, but internally, it has crossed a universe. The genome is assembled, the first division complete. Totipotency is at its peak. the foundation of every future organ system, the nervous system, endocrine system, circulatory system, has been conceptually drafted at the level of genes and molecular gradients. Life has not yet taken shape, but its architecture is now set in motion, cell by cell, signal by signal, in a darkness deeper than the ocean floor. For all its complexity, the first 24 hours of life unfold in complete silence. No senses, no consciousness, no awareness of the world outside that microscopic chamber. And yet, within this darkness, the body begins shaping its future with a precision it will depend on for the rest of its existence. Two cells that began as strangers now operate as a single unit, their chromosomes aligned, their instructions unified. What was once potential is now direction, a blueprint not just stored in DNA, but carried in gradients, in ions, in the quiet hum of metabolism. Each division, each rearrangement is a message. Continue, refine, protect. This is the essence of early life. Not spectacle, but subtlety. The embryo doesn't need complexity to begin. It needs coordination. It needs the steady internal logic of biochemical signals passing between membranes. The earliest form of regulation resembles a whisper between molecules, a prelude to the nervous system that will one day interpret the world beyond the womb. The cells move forward without hesitation, not because they know what they will become, but because the architecture of life has been built to proceed. Every organ, every neuron, every future thought begins from this choreography of division and restraint. These early blastomeres do not specialize yet. They hold every possibility quietly, like unopened pages in a book waiting to be written. It's easy to imagine life beginning with a heartbeat or a breath, but here, in the first 24 hours, life announces itself through precision, through the flawless pairing of chromosomes, through the stabilization of a genome, through the first committed steps toward becoming something more than a single cell. The world outside continues unaware, yet inside this silent chamber, a lineage unfolds. Generations of inherited information converge to create a singular combination that has never existed before. This is not just biology. It is continuity. It is resilience. It is the body demonstrating from its very first moments that growth is built on connection and cooperation. As the embryo prepares for its next journey, down the fallopian tube toward implantation, the momentum of life is already irreversible. The first day has passed. The foundation is set, and from this point onward, every moment of development will be an expansion of the spark lit here, in darkness, in stillness, and in extraordinary quiet brilliance.